Okay, and now I turn the table to you guys. You guys, why don't you first introduce yourselves and then get started?
Sure.
So I'm Reza. I work at ABL. I'm a bioinformatics analyst, and that's it. We work with a lot of labs.
Yeah. I'm Joe Pucella. I'm in the Reza lab. I'm not a bioinformatician. I'm kind of bringing the biology aspect to this. So we're gonna tag team.
So yeah, so I guess we're gonna have it like a conversation based. So I'm just gonna code. So we developed this tool called iCellR. It's a tool that is capable of analyzing single-cell RNA sequencing and single-cell CITE-Seq and single-cell VDJ sequencing. And Joseph from Reza's lab, from Force's lab, they helped us with CITE-Seq, and Nicole was here. They from John Prucci lab, they helped us with single-cell VDJ single-cell sequencing, some of the concepts and ideas that we added to in terms of functions to the tool. So if you Google iCellR, I stands for single cell and R package, you would be able to go to the website. The manual of the tool is just like any regular R package manual. You can see the functions in there and how do they work.
So maybe you should mention that this isn't, so iCellR is not specific for CITE-Seq, but this analysis will help us get into transition into the CITE-Seq technology.
So yeah, you can also analyze actually bulk RNA sequencing too. If you have a lot of samples, bulk RNA from TCGA downloaded from other places, you can use iCellR in there too, and I can show you some of the data from TCGA as well in the end. So today, where's the code? So today we're gonna just analyze one single small CITE-Seq data, but in the end I will show you a couple more already analyzed data too with iCellR. So after installing iCellR, you can load, if you're familiar with R, you can load the package, and once it's loaded, you can go to the directory where your files are. It's taking a little bit of time. So if you have a sequence with 10x genomics, there's a function called load 10x, and then you can just direct it to, if you know where, if the files are right in there, you can just use this function to read the data. It can read zipped or unzipped files, and it works just like this.
So Reza, what's the data you're going to be analyzing today?
This is
My data set?
whose data set is this? This is don't remember whose data set is this.
I mean, like, what are the cells? What are the?
So these are blood. These are blood samples. But I would remember it just like it's in the top of my head. But this is just a small proportion of someone's data that is kind of like already published, probably. So we have, not probably, it's published. I'll talk about that. But we have some of your data already analyzed too, because yours was bigger, like about 40,000 cells, right?
Yeah.
We didn't want it to. So this is just a small.
The antibodies will be.
In this one, I guess there's only few, but I have a couple of other data. One of them was 70, 80 antibodies. Yours is how many?
My data set has 81.
81. Yeah.
antibodies,
Yeah.
So,
Did they all work?
No, they don't all work. So if you run into
Yeah.
good news then.
I mean, technically, if you optimize each and every one, it should work, but it's a lot of work to do that. So, we use some antibodies that we know work in flow cytometry and have been optimized for that. And we just go ahead, and then we can tell if it doesn't work, we toss it out. No big deal. We lose 1, 2 antibodies. Out of the 81, 76, 77 worked.
Oh, not bad.
Yeah, it's a
We had
good.
one with Atlantis lab, and it was after doing a couple things, it was working well too, right? Right. So I don't know how many they had, 60, 70. It seems like most of them work. I don't know.
These antibodies was the CITE-Seq, the total Seq 8?
No, yeah, so this is one of the commercially available from BioLegend, Total-Seq. We actually conjugated all of our antibodies in-house with our collaborators. It ends up being cheaper to do it that way, but it is also commercially available. And I think BioLegend offers over 100 different ones now.
So yeah, so once you load the data, you first load the RNA-seq data, and then you would load the ADTs later. So this is just generally how it looks like. I'm just printing the head of the data and the first 5 columns. So here you have the genes, and here the cells, right? So if I do, if I actually do the dimensions of the data, this one we would be able to see how many rows and columns. This is, yeah, actually it was in there, 2,700 cells and about 32,000 genes. So we're not going to analyze this one. I just was showing you how to use the load data, but there's another data that I've already loaded, and this one has about 4,000 cells, and I just already loaded it here. But I'm going to take a portion of that too again. Then let's see how big is this one. 8,000. So if I just take, let's say 2,000 for now, 1 to 2,000, to just be fast. So now that we have the data, so it looks fine. After having the data in this format, I had it in an object called RNA. I'm reading the ADTs as well, and I'm going to name that as ADT. And here it is.
So ADT stands for antibody-derived tag.
So these are your
the
CITE-Seq.
CITE-Seq signal.
Yeah.
Do I make the windows and text a little bit bigger? 'Cause it's hard to
Sure, of
see
course.
that.
Maybe I can put
Yeah, close in the back too.
Yeah.
Minimize
this one's better.
No, they're like black and green.
Yeah, I'm gonna make the transparency go away. I wasn't prepared for this. Well, I didn't prepare well. Where's the? I'm gonna just take this out so it's not like that.
Yeah.
Is it clear enough?
The font is just really small.
Okay.
Can you just command plus?
Here's the word. Do you see? Preferences. That should be out here. Profile, right?
Guys, I think you press command plus in the channel.
plus is it?
Oh, and this one?
Yeah.
Oh, there you go.
Yeah, it's good. Yeah, it's better. Okay. So yeah, we have the ADTs here, CD3, CD4, and here are the values for the first 5 cells. And then the next thing you need to do, since you're analyzing with our tool called iCellR, you just make an object of iCellR, class iCellR. And the object looks like this. You just make object and then read the RNA, and then once you did that, you can just print the object to see how it looks like. So it just tells you it's a class of iCellR object. There's 2,000 cells in it. There's no condition in it. It's just one sample. You can have multiple samples. These are the first few gene names, the first few cell IDs, so you can just see the structure of the data. There is no QC performed, no PCA performed yet. There's no CCA performed. And as we go along, these will be developing. And then here's your CITE-Seq data. We haven't loaded that yet, so this is empty. And if you have BDJ data, you could add that from here too. So to run a QC, there's a function called QC. This uses a bunch of genes that are already in the default, but you can use your own gene sets for S-phase, for cell cycle genes and mitochondrial genes. And doing that, you would be able to create a plot like this. And if you have more, okay. Doing it.
What's this?
It's pretty slow.
That's the transcriptome?
Oh, yeah. That would be faster than this. If you have multiple conditions, you would have multiple ones for each one of these. So this is your number of genes per cell. Each dot is 1 cell. This black dot is the mean. This line is the median. So there's a box plot, and then here's the violent plot around it. Here the number of UMIs per cell, and here's the percentage of mitochondrial genes per cell. And then here is the S phase probability per cell and G2 phase and N phase. And then later we would see how we can convert those to actually find out what cell cycle each one of these cells probably are assigned to. So after doing that, you can perform a couple of other QCs, maybe not so important, but like they look like this. If you just wanted to see how your data looks like, if you had more conditions, it would be more useful to see if there's like so much of a difference. In this case, there's a percentage of mitochondrial genomes versus UMI, so it's just like scatter plot if you wanted to do that. There's a bunch of other things you could do with this stats option. If you wanted to know more of the functionalities of the each one of these functions, you can just put a question mark in the beginning of them, and just like any standard R package, you would be able to see what are the parameters you can put in there and what you can do with them. So that's that, and then you can filter your cells if you wanted to. So in this case, based on that plot, let's say if you want to filter any cell that has more than 10% mitochondrial genome, less than 10%, and the coverage would be anything more than 500 genes covered. You can play with that, have 200 or anything, depending on your data. So we're just gonna put these filters just like this, and then this would create a file in there telling you what are the filters you put in there. So if you wanted to do assign your cell cycles, you can just run this cell cycle assigning algorithm, and that would quickly, you can actually, if you look at the head of the quality statistics, you would be able to see, okay, this cell number 1 is a G phase cell, and then you can plot that as well here quickly. So this is typical for blood cells. This is what you're supposed to see. If you had cancer or other cell lines, you would see a lot more dividing cells. So, and then for the normalization, there's a bunch of normalization methods that are in here available for you. You could just quickly check. There's DE-Seq. You can choose from DE-Seq, rank DE-Seq, global GLSF, rank GLSF, read per million, is spiking, and if you don't want to normalize your data and it's already normalized somewhere else, you can just choose not normalize or just have the data, just don't run this function. So we're choosing the rank GLSF based on the top 500 genes, normalizing the whole data. This is just GLSF stands for library size factor. Global library size factor, and this is taking the first 500 genes to normalize based on those. And here, since we filtered the genes, cells that not having more than 500 genes in them, so the majority of the cells would have coverage more than 500 genes. So it's safe also to put this top rank as something that you could put as in here. It seems to be giving a better result. So once after doing that, you can just run a QC on your genes to see how your genes look like. It's gonna be very fast. So here, for example, this gene 1 is expressed more than 0 or 1, like only in 2 cells out of 1,800 cells. So there's 1,800 cells after filtering. There was originally 2,000 cells. And this is the percent of the cells that express that gene, and this is the mean expression over all the cells, and here's the standard deviation over all the cells, and this says in all the conditions, although we have only one condition here. So, and if you look up again your object, you should now say that you already performed QC, but PCA or CCA is not performed yet. If you have more samples, you could do CCA as well. So here, quickly, we can just run a plot that would show you the dispersion of the genes and how the data looks like. So we would be using these green and blue genes. So every dot is a gene in here to perform PCA. And this you can design it however you wish. So this black line is just the dispersion limit that we put in here, 1.5. So, and this red line is the line that we chose for, so these are the mean, the expression, the genes ranked by their expression. So here we're choosing the top 500 genes. So the idea is, so you could just choose the genes that are dispersed to perform PCA, but you could also just use the top genes expressed where you can have a combination of those. And this would create actually a file with genes in that in the folder that you're analyzing your data. And it says that this file is created my model genes. And then you could just go in there and choose your own genes in there and modify that, the gene list, and then perform your PCA based on the gene list that you like.
Can I ask you a question?
Yeah.
This is all done on the RNA, like a single cell RNA-Seq data, right?
Yes.
We didn't touch the CITE-Seq data.
Yes,
so
we're
far.
going to.
And we need to do all this like normalization and everything for the CITE-Seq?
Yeah, well,
Yes.
all this needs to be done also for the CITE-Seq data on the transcript, on the transcriptome. But you know, you could, in theory, you could do clustering and everything on the ADTs as well. But it's
Yeah,
not
you could
the
do that
typical, not the typical pipeline.
What is better, to do it together or?
Yeah, I mean, you would.
I think the best is
Yeah.
to cluster your data on RNA and just overlay the ADTs on them to see how it looks like. Usually they correlate. We've done it the other way too. Do the analysis on ADTs and then overlay RNA on it. That sometimes works too also.
Yeah, but typically what I've seen and what I've done also is everything up to
Everything based on
clustering
RNA.
based on RNA, and then you have all this CITE-Seq information that you can overlay on those clusters and decide how to move forward for the next iterations of the analysis.
But did I miss it? So do you normalize the antibody data?
Yes, I'm going
Oh, you're
to
gonna do that.
add the antibody data and normalize it, and that's normalized a little bit differently than this, and then merge the data together.
Maybe we can skip some of the-
Yeah. So here's the normalization for ADTs, and then so actually after normalizing it, since the ADT data is very small, it should be very fast. So here it's already done, and
ADT is the antibody?
antibodies. Yeah.
Yeah.
So this is before normalization, how the data looks like. Wait, what happened? Raw ADTs. Oh, we haven't looked. Yeah, we haven't added the ADTs to the object yet. So that should be add ADTs. Did I have it here already? Yeah, add ADT. So here you already read the data, ADT data to ADT object. Here, if I skip that. Now it's added to the iCellR object. If you just look at it right now, you have your raw data, raw ADT data now already updated there. So now after normalizing it, here we are. So if I normalize the data, and here's how the data looks like. So the one in the bottom is before normalization, the one in the top is after normalization. You can tell they're normalized because they don't have their integers here and they have decimal points here, right? Usually.
What's the normalization?
So this is just global size factor normalization.
Like mean?
Sorry.
Mean normalization?
No, it's based on the library size. So it's the
Like
TPM.
TPM.
Yeah, but the only difference is that here we just transpose the data. So here it's just, because every cell only few antibodies, we do it per antibody count
Oh,
every cell. So it's just the same, it's just like transpose. So that's that.
So the sum of each line, the sum of the lines will be the same
Yeah.
and not the column, like
The sum of the so the
Like
columns
when you
would be
do
genes.
TPM, the
Yeah,
sum
the
of the
columns would be
columns are
genes,
the
so
same.
yeah.
So now the sum of the lines are the same, like you assume that each antibody was represented the same number, like you normalize to the same number.
Yeah.
Okay.
So and then once you do that, you can just add your ADTs to merge it with the RNA-Seq data. here we are. Okay, here we have it normalized, and then here's all the CITE-Seq data done, right? So, oops, okay. So we have the gene list in there. You can just run your PCA analysis. And here for the PCA, we're just choosing the genes that were in that file that was created earlier. It should be fast. So this is only 2,000 something. And if you wanted to, if you have only one sample, sometimes with more samples this works too. If you wanted to, you know, once you run PCA, you have, you know, all of your cells clustered. But if you want to kind of like even have them more separate, you can run this function. This would find, so you would say, okay, I want to only look at the top 10 clusters, top 10 PCs, and then take the top 20 genes, or you can change these numbers, top 20 genes for each PC, and bottom 20 genes, or 5 genes, and then rerun PCA again on those. This kind of, so these genes would be automatically, if you further analyze data, when you do the marker genes, finding marker genes for each cluster, these genes would already come up there. So you can just earlier, you know, choose them here and then just run PCA one more time if you want more segregation, like kind of have each cluster to be a little bit further far from each other, more defined if you want it, but not necessarily. So if you wanted to run this, you can run it two-way, two-step PCA. Here for clustering, so again for clustering, there's about 30 indexing methods that are in there. So you can choose from these methods. There's RAD-D, RAD-D2. There's k-means and median, centroid, and from the distance analyses methods, everybody uses Euclidean, so the default is Euclidean too, but if you wanted to choose anything else. For indexing methods, there's about 30 methods in there, and there's pros and cons in each one of them. Some of them are very slow, some of them are fast, some of them tend to be more sensitive, some are more stringent. The default is Silhouette, but KL is good. There's a bunch of them that are good in here too. So this indexing methods would be just walking through your data and then tell you like what parameters would be good to find what is the optimum number of clusters based on your data. So there's a variety of things you can do here, and then you can choose how many dimensions of PC as well you can use for clustering, and you can also put like maximum number of clusters, like don't find more than 25 and don't find less than 2. This is the default. Usually, if it's something more, it's close to 25, it's better to just increase this number. So if there's possibly more than 25 clusters, you can just see that. So this is, since there's only, maybe I run something faster. Okay, let's do this. So look, this is a little bit slower than the other ones. Okay.
So I asked a question following just now on your talking about the normalization of antibodies. So what's the rationale of normalizing by rules, by the antibodies?
So if you have, like the columns as cells, and you have only 5 antibodies, right? And you count them all, summarize them, right? And sometimes they're not captured so well for each cell. But if you just count all the antibodies, you could do it the other way too. But this seems to be working better. So, but if you count like all the values for one antibody across all the cells, and then get the sum that way, it seems to be working better.
So this is practically what yours.
Yeah, I mean, yeah, I've done it this way every time. It works so much better than the other way. So,
So without normalized to something like a hash antibody, that's kind of considered to be a constant, or
yeah.
especially antibodies?
So And so the hashtagging is already done. So we just got the cells and or the conditions. So
Well, I mean, just as a normalizing concept, like if it's good to work, if you want numbers, you just say, okay, more sense to look at something that's working with the antibody and using that.
can you speak a little bit about?
I guess I still have an issue with understanding how this one works. I would just assume that you would normally normalize to something similar, like an antibody. So like an antibody that binds to everything and then compare the other antibodies to that. That's what I'm asking.
Another possibility actually is you can also put in isotype controls, which might make sense for some cases. I haven't really looked into which ones perform better, but we have isotype controls.
And some people don't even normalize the antibodies because I don't know, and that works too. To be honest, I guess CIROT they just log transform them. So that
Which is basically what you would do in like a flow experiment, right? You wouldn't normalize it to anything. You would compare it to how it looks to an isotype control, but you wouldn't necessarily normalize your signal. So I'm not.
Yeah.
I mean, normalizing might not even really be necessary.
maybe for
altogether.
antibodies, yeah, but.
But I guess it makes sense if later, for a given cell, you want to look at like 2 antibodies, but they each have
Yeah.
2 very dynamic ranges. One is like really highly expressed. Then what's interesting for you is kind of like the fraction,
Yeah,
and then you're essentially comparing fractions.
Yeah.
Like,
so we figured out like if you transpose the data, so have the antibody in all the cells and sum that up,
yeah.
it seems to be working better, so. If you don't normalize them, it should work too, I think, and a lot of people do that too.
So just not normalize at all.
Yeah, yeah. But then we run imputation after, I'm going to talk about this, and then that really makes a big difference. It makes, some people also have very low antibody capture, and it just like makes everything, your heat maps look so much better, and antibodies like, precisely just show up. And I'll show you a couple of data on this too, before and after imputation.
You take the CITE-Seq data?
Yeah.
Or you impute it?
So once the CITE-Seq data is merged with RNA-Seq, we use RNA-Seq data for imputation, and then this CITE-Seq also gets imputed. Imputed. So that seems to be working very well. We just did that as an experiment, and some data that everybody thought it's not good. They thought it's just we need to sequence again, and we did that, and it was like every single antibody worked so well for each cluster. So that's just an idea if you wanted to do. So the clustering is done. So then you can run PCs, t-SNE on the first 10 dimensions, or however you can choose some parameters in there. And then run UMAP and diffusion map. Since this is a small, I'm going to just run these 3. And maybe let's just do, t-SNE is the slowest one.
Maybe just do the UMAP instead of
Yeah,
all of them, because we don't have too much time.
Yeah, because we can show other data too. So so that's done. I'm going to just show you maybe the UMAP plot. And the good thing about iCellR is that all the plots in here, you can, so this is not probably well. Let me show you the real data with all the 8,000. You can just look at.
What's wrong with that?
This was good too, you can see. Good. Okay, so there's, how many clusters now with the parameters we put in there? About 11. So, yeah. So once you do this, you can look at your, you can impute the data. From here onward, I'm just going to load the data that we already analyzed, because some of these are slower, and then it would show you what the data looks like. So if I just load the already analyzed data with the 8,000, all the 8,000. And then we can see some other things that you can do much faster. Okay, while that's loading. So here, if you run all the, UMAP and TISNE and diffusion map, this is one way to look at them with the TISNE. So everything you do in iSellR, there you could also have a, you can import them in a, how do we do this? So you can have them in an interactive format as well, and then you can zoom in, zoom out, take pictures, and you can, mouse over every cell and tell you which cluster it is and the
You can
value.
show or
here.
hide different clusters.
Yeah, as
That's
well,
really useful.
you could also do, since you can also do cell gating, there's also 3D if you wanted to look at 3D plots. Because sometimes if you have a 2D and it looks like this and you think these two are overlaying on top of each other, but if you turn it around, there's distance between them. Sometimes that's useful too. These would be just plotting your data. Diffusion maps. And if you had more conditions, you could also color them based on conditions. And then if you wanted to make any interactive plot, you can just choose this interactive option as true, and then give it a name, and it would make an HTML file. And you could choose from clustering by conditions or clusters. Plotting by clusters or conditions, and choosing from TISNE, UMAP, diffusion map, or other things. So that's that. If you want to cluster your plot, and then you can find markers. So this would go ahead and make markers per each cluster, and then the result of that, if I read this one, it would generate a file like this. If I just hit it, generate a file like this. So for example, this gene number 1 is a marker for cluster 1, and this is the base mean average across all the cells, standard deviation across all the cells, average in cluster 1, and average in the rest of the other clusters. And this is the fold change, 300 fold change. This is the log 2 fold change. Here's the p-value, adjusted p-value, and here's the average for each cluster. So you can see, cluster 1, the average expression for that gene is this much, and you can see other clusters as well if you wanted to. And then once you have your marker genes, you can choose, if you wanted to take, I want to just top 15 genes from each cluster, and then I want to put a minimum of base mean of 2. You can do that, or modify that. So here you get some genes for each cluster as markers, and then you can quickly heat map them. The heat map would look something like this. Oh no, this is the ADTs. Something like this here. Right. So cluster 1, these genes. Cluster 2, these genes. Cluster 3, these genes. So the genes that come up as markers. And these are the top 15 genes. And this is just the sample data. I'll show you other data as well. And after imputation, if you wanted to impute data, it would look something like this. So the zeros would be filled. So since the 10x genomics, the coverage is low, a lot of cells won't be captured in the sequencing method, so it would be zeros instead. So you could impute the data. There's a lot of methods to impute the data. I guess you guys do that too a lot.
We do that too.
Yeah. So.
I'm wondering if you could skip ahead a little to the CITE-Seq though.
Yeah. So this is done. So CITE-Seq is ready. So you could when you do this, the marker genes, it would also find the CITE-Seq data. So it's already analyzed too. So you can just read your CITE-Seq ADTs. Here I'm just reading them right now. So these are your CITE-Seq IDs. You can heat map them as well. In this case, not all of them have worked. So for example, this one is CD19, which is a marker for B cells. You could look at that. I'll show you other data that have worked much better too. And you could also do predict cell types with iCellR.
So each column is a cluster, or
Yeah. Each column is a cluster,
Or weren't
and these
there-
are the ADTs.
there were 11 clusters, weren't there?
This is
Different?
this is the other data that I
Okay.
So There's seven. So you can see this one is only specific to cluster 7. Some of them are only in 2 clusters, like CD3, right? This one is higher, and this one is only for one of them. Here's another data. Maybe I can show a little bit of stability.
So when you said that some of them didn't work, what does that mean?
Some of them that didn't work
like-
would be like this one.
Like it's not specific for a particular cluster?
Maybe. Actually, these seem to
The
be.
last
For
one?
example, this last maybe this also seems to be good line, right? CDA.
I've seen in my
Yeah.
data set, there's a couple where the signal is just across the board too low, not
Yeah.
above the background.
So for
that we
example-
just chalk it up to.
Yeah. So
work.
for example, this is. Yeah, sorry. This is CD3 ADT. It's in here, and then here's the CD3 gene, CD3E. So they're overlapping, as you can see. So CD3 antibody worked well.
Yeah, I think this is also a really important validation method as well, because you go into these experiments a lot of times knowing what you're looking for at least certain markers, and if the transcript and the ADT line up, it gives you confidence to proceed with that antibody, right? If they don't line up in instances where you know they should, then there's potentially a problem with that antibody.
I just think it's kind of like ironic that we use these antibodies as kind of like they're supposed to validate. We see like we can't trust the transcriptome, we want to validate it, but then we look at the clusters generated by the transcriptome and we say it worked if it matches with our transcriptome. So the transcriptome. So what do we believe, the transcriptome or the antibodies?
Well, I mean, I think if they match and they match your expectations, it gives you more confidence, no matter
If they
what.
line up, they agree. Because it might be interesting to take like all the cells that are positive for one that may have looks like it doesn't work, and see which transcripts are high and cells with high for that antibody.
Yeah, I mean, I think this is also really the main strength to me of CITE-Seq is that you can go back and forth from one data set to the other, whereas in the past you'd have to do separate experiments altogether, right? Here it's all integrated together in a nice data set already. So, what might have taken weeks to go from sequencing experiment to flow, back to sequencing, back to flow, is all in one package.
Right.
That to me is the biggest strength.
Yeah. And also is gating too, right? Sometimes useful.
Well, yeah, I think the cell gating, I don't know if you were going to
Yeah,
show an
I'll show you
example of
one
that,
gating
but
too.
the cell gating, I think, is a unique thing that iCellR offers that really takes advantage of this CITE-Seq data.
Because Itai talked about it too. There's no single tool that would do everything for you, or single algorithm or clustering method. You would be able. You need to play with the data. Sometimes some people say you need to beat the data until it speaks.
Yeah.
And that's why people come to experts like Itai and other people just analyze our data because they have experience, or we have experience with sometimes you need to just work on the data so much, and none of the clustering methods sometimes don't work so well. And I'll show you some of the examples. And sometimes you just, as an expert, probably just want to gate around, turn on a gene and gate around. It only turns on some genes,
Yeah.
some cells, and then just gate around and say, okay, this is my cluster number 19
Right.
or 20. Or,
Right.
Yeah, so I guess to bring it back I don't know if any of you are immunologists, but we're very used to working with flow cytometry data in FlowJo or in some other application similar to that. And what I think is really intuitive about this package is there's an option where you can do cell gating just as you would do in FlowJo, right? So if you are looking at CD4 T cells, normally you gate on TCR beta or CD3 first, and then you'd go CD4 positive, CD8 negative, right? The unbiased clustering, while it should, may not always give you a nice clean population. But if you're really interested in just that population, you can skip that and just gate it as you would in FlowJo. Now gate the CD8 T cells, for example, and you can compare those transcriptionally. So it's almost like a hack in a way, where you can use unbiased clustering as well, but if you really want to hone in on something, you can also do the manual cell gating and then take advantage of all the transcriptome tools at the same time.
Right.
Also, you could do gene-gene correlation. Like in when you do cell sorting. some people do CD4 and CD8, you know, you could do the same thing after imputing the data. And then, so these cells are like CD4 positive and 8 positive, and then just choose those based on APTs or even genes sometimes. Or like just see if a gene is, you know, like for example, I have a couple of examples I can show. So here, let me see if I can find those. This is maybe not in here. Probably put it somewhere else.
Just to continue with this cell gating function, again, it's very intuitive for me because I come with immunology background. But there's also examples where I've seen the clustering gives you this cluster that seems to be a little bit split, whether it be on the Tisney plot in particular, or even just for expression of a certain gene you're really interested in. Sometimes you see a gradient across a single cluster. Now, if you want to study the side of the cluster that's low versus the side that's high, you'd have to come up with parameters that would split that cluster first, and then you can compare them more easily to do differential gene expression or something like that. With the manual gating, you can really just draw two circles, right? And now you can compare those two poles of the same cluster in one step.
Or something like this.
Speeds things up.
So we had cluster 7 that we already looked at in this plot. Cluster 7 here. So here, cluster 7 is this pink one right here. And that's NKJ7s, natural killer cells. That's natural killer cells. So if you do GNLY as a marker for natural killer cells or NKJ7, you would see that cluster 7 has the highest for both of these. And then, you know, even if you don't want to cluster or like do Tisney or something, if you just do this, you can say, okay, these are my natural killer cells. You know, just put, if you have some genes, you know, or CD4, CD8, and just say, okay, this is my, and then say, okay, up to here I trust these are my natural killer cells, or bottom ones are not. You could do something like that too as well.
Yeah, and this is really very
And then
analogous to Flojo.
Yeah, and it would tell you the correlation of these two genes across all the cells, or you can just choose one cluster, or have it interactive and just choose one cluster and see the correlation of those p-values of those. You could do something like that too. And one of the other unique things, I guess, that iCellR offers is that it does cell prediction. Why this doesn't close, I have no idea. Okay. Okay. So, so you could, for example, here, if I say, okay, that's what we want. So types. Okay. So here, let's say you want to choose. Let's say you did this. You ran this function and then found top 15 genes for each cluster, right? Come on, it's not running for some reason.
Top?
Yeah.
What makes them
So these are like sorted by the fold change and p-value. So you know, the good genes come up for each cluster. Okay. So let's say you want to find the top 50 genes in cluster 7, right? Or cluster 1. I'm sorry, these are freezing for some reason. Okay, let's say cluster 1, right? So now you have the top 50 genes for cluster 1, right? And then you can choose mGen's data and then do a heat map, or even better yet, you can do a point plot, right?
But we're kind of overlapping a little bit.
Yeah.
Okay, oh, you're overlapping?
Yeah.
There's some things that we've spoken about, right?
Yeah. So as you can see, the top predictions are coming as B cells, and we already know that cluster 1 is B cell. Or you can use mGen ultra low input, which has 157 samples, and then this would also tell you, oh, this is the heat map of it. You can see the B cells are. I need to make that bigger. B cells are all red in there. Let me do the point plot, which shows it better.
Yes, I think this problem has come up a few times already in discussion about how to identify what a cluster is, what population it is.
Yeah.
You know, you can always go through and look at individual markers by eye and kind of do this in a supervised way. This, I think, is really nice because we are always asking, well, you know, what's an unbiased way to assign what these clusters are? And this really is the best tool I've seen to do that. So this takes a lot of the guesswork out of assigning clusters to me at least.
So as you see here, that all the top predictions are coming as B cells for cluster 1. And if you do cluster 7, we know they're NKJ7, they become natural killer cells. We also have tissues from mouse cell atlas, and if you do that too, if you have other tissues than blood, it would kind of predict which tissue the sample is coming from. So in this case, I don't want to do heat map, I want to do the point plot. So in the heat map, you would be able to see these genes that you've chosen, the top 15 or whatever genes, and then see which tissues express them highest. But if you wanted to do a point plot, it would tell you, okay, for example, we know they're blood, so cluster 1 is blood, so it kind of comes as spleen or blood in this case.
So this is really cool, and I think really shows how powerful iCellR is. I want to give Joe an opportunity to show slides if there are some things that
We could, yeah, there
wanna
probably
switch?
should only be about maybe 10 slides that aren't things we already spoke about. We can go through that quick.
So maybe do that now.
Yeah.
But before, I just want to give a plug. Prateep Chattopadhyay wanted to be here, but since he's not, I have to do his commercial for him. So he's the director of the. Sorry, he's the director of the Precision Immunology Core, and okay, yeah, now I can do it. And there's a method there called AB-Seq, AB-Seq, and it's not transcriptomics, but they get 400 transcripts, which is a lot, and also he does 102 antibodies. So I haven't used it myself, but if you have samples where you want to characterize 100 antibodies, go talk to Prateep.
Yeah.
It's Precision Immunology Core.
I'm going to just pass to
And then
you. So
we're
just
gonna pass it over to
going to
Joe.
show a couple of images. So this is just, if you wanted to gating, you just gate around this, and then you download your cell IDs and then just rename them in clusters. So in the
Show the live like video you have on the top. I think that's
that's
cell gaming.
Yeah, you could just like choose your cells like this.
Because that really shows how
Yeah.
similar it is to FlowJo. It shows
then-
how powerful it is.
Yeah, and then you can download those IDs and then rename them cluster 20 or 1 or whatever you want them. There's, you could do also this if you have more than one sample. For example, in cluster 1, all the 3 conditions are kind of normalized, and you can tell you like which sample has more in each cluster. A few other things you could do, these box plots, bar plots, everybody does. But a few other, you could do pathway analysis with KIG, and then if you wanted to look at each cluster's QC, for example, mitochondrial expression in each cluster or gene, coverage in each cluster. And one unique thing about the differential expression analysis in this one is that you could say, for example, I want to compare cluster 1 and 1 between wild type and knockout, or cluster 1 and 2 only in the wild type. So you could choose a combination of different possible ways to do differential expression analysis and do Volcano plus and MA plus. These are also interactive. You can mouse over and see the genes. You can also do pseudotime with it too. And we're playing with some versions of diffusion maps. That kind of keep pseudotime trajectories and stuff. That kind of looks like snakes.
Whoa.
That work, yeah. If I have a better, so these are developing and soon coming, kind of this or kind of these. And there's also, if you wanted to have it more simpler like this, then you can do that too. So,
Very artistic.
yeah, and or something like these guys or these guys.
Can I ask, the package also works for hashing or only for CITE-Seq?
So we have a function for hashing that would predict based on percentage each cell would be from which condition you have, and then you can use that. It works fine, right?
Yeah, I have a slide about that
Yeah,
also.
You wanna?
But hashtagging with CITE-Seq antibodies, I'm not really sure about other methods.
It's just like the hashing is like, you know, it's like on and or off. It's not like an expression level like the CITE-Seq barcode. Like it's, is it there or not?
Yeah, well with
But
CITE-Seq
you don't really
it's...
get the data like this. Like the data is not 1 or 0.
Yeah,
and you want it to be. So.
Right, yeah.
Yeah, I can show you
that's a
one.
good point.
Yeah, if you have a slide on that.
There should be something in there. I'm going to just go through fast. Some of the slides we've talked about already, but you know, as Itai already mentioned, so CITE-Seq allows you to look at the surface phenotype at the same time as the transcriptome using this specialized antibody that has a barcode conjugated to the antibody.
Can I ask you a question?
Yep.
So, just technically, you just take your pool of antibodies,
Mhm.
right, and you just throw them on the cells, you wash, and then you start the RNA-Seq.
Exactly, yeah, so it's schematized right here actually. So the only thing that's different is really just that you have to stain with your pool of CITE-Seq antibodies in the beginning.
Right.
And then you put them through, for example, the 10x chromium controller,
Uh-huh.
the same you would for any single cell RNA, or you can do drop seed also, whatever.
So do you have any trouble with the barcoding? Because you have 2 sets of barcodes now.
No, I mean,
Never any issue, technical issue?
no,
Because
I
it's
mean,
on
you decide
on
what.
the other read. It's not on the same read. The barcodes.
Yeah, I understand
Yeah,
that.
it's,
Yeah,
I mean,
I'm
it's
just
all
thinking,
things that you decide ahead of time, so.
Yep.
Do you need to do any blocking?
You can, yeah. I mean, you mean FC blocking or what?
Yeah, I mean, is it needed or no?
Yeah, I did FC blocking on my samples. I guess it also depends what your samples are. If you expect that you want to see FC receptors, you may need to, you know, devise certain conditions specific to that. My samples, we did FC blocking on all of them. Yeah. So, I mean, this is just a schematic, but it's essentially the same as single cell RNA-Seq. This is the main step that differs, and it's analogous to a very simple staining you would do for flow cytometry. And then you, when you do library prep, actually the transcriptional data and the ADTs end up in different libraries, which is also an advantage if you need to manipulate these differently. Okay, I think we talked about a little bit of this already. I really want to emphasize though that integrating these two techniques together it's hard to explain how powerful it is, but in the past these would be experiments you'd have to do separately, back and forth over weeks or months. Now in one experiment you can save all that time and have the data in one shot. And it's really powerful for if you want to, for example, identify novel markers you'd want to use for phenotyping, or for example, we work with bone marrow progenitors a lot. So if we want to find a progenitor that is common to 2 populations, we might be able to identify that using the transcriptome data. But then to actually isolate them, we still need a surface marker. So if we have a pool of 100 surface markers already in that data that we know work, it makes that whole process much faster. Okay, it's really only limited by the availability of the antibodies. It's not like flow, obviously, where you're limited by spectral overlap. You have enough barcodes to do every protein in the genome if you wanted. So there's no limit. Again, cell hashtagging is a good feature with this. So what this means, I think I have a slide on it actually. Yeah, I do. I'll skip this. This is just to show that the data looks similar to what you'd expect by flow cytometry. This is the list of markers that I use for my latest experiment. If you're interested, we can look into this a little bit more later. Okay, so this is something that seems like people were interested in. So the idea with the hashtagging is that you have, let's say you have 3 samples. In my case, we use CD45 to include this hashtag information. So on my 3 samples, all the cells have CD45 expression, and we give a CD45 conjugated to a different barcode for each of these samples. So the real advantage here now is you can pool these samples together and run them together and save money and resources when you're doing the experiment. You can put these all on one lane of the chromium controller, for example. And then afterwards, you can, demultiplex this and figure out which condition is for which cell. Okay. But it's not strictly necessary to use CD45. In my case, CD45 is the most obvious, but in other experiments, you can tailor this to whatever might work the best for you. And I think in our iCellR, you have a function to demultiplex this.
Yeah, I can show you.
Right,
I can show you some part of your data.
Yeah.
How it works.
Joe?
Yeah.
Are the published protocols for how to conjugate your own antibodies? Or-
Yeah. Yeah, so the developers of CITE-Seq have made all of this publicly available. It's not in the original publication, but if you go to their website, which is this link on the bottom here, it's just CITESeq.com. It's really easy. They have all their protocols listed there. It's very transparent. So another feature that I think is really useful actually is this doublet removal. So you can, I think it's becoming a little bit more clear now that when you run these single cell experiments, you may have contamination from some doublets that don't get properly separated in the early steps. And in the past, there's no way to really discriminate these and get them out of your data. But if you do hashtagging, for example, like with CD45 barcodes, you can actually very easily get rid of these doublets, and it cleans up your data significantly. At least it has for me. So the idea basically is any cell that has barcodes for more than one CD45 that you stained with these in the beginning, before you pooled, you would omit those because there should be no cell that should have antibody from both. So it implies that your lipid droplet contains cells from two different samples. Okay. And there's just a couple of things I was going to show here, just to show validation for my experiment that certain antibodies really look very, very clean. So on the left is the ADT signal for a marker we use for plasmacytoid dendritic cells, sigma H. And on the right is the transcript for the same marker. And you can see very clearly that they are almost perfectly overlapping. This is just another marker that I use often as well, BST2. And again, you can see that it looks really, really good here. It is worth noting, though, that there isn't always perfect overlap. And this could be technical reasons, or it could actually be for biological reasons as well. In this case, at least in my experience, CD4 transcript doesn't always work very well, as from what I've seen. I'm not sure if this is a common thing other people have seen. So typically the CD4 transcript data isn't really useful. In this case, because we had the CITE-Seq antibody for CD4, we have information for this marker, which is typically a pretty important marker in immunological studies.
And you anticipate that all of those cells should be CD4 positive? Is that right?
So it's a little bit hard to see the gradation here of the red, but these should be the strongest, and then the rest are a little bit lighter.
Yeah, so a lot of background.
Yeah. And again, you can, between the imputation and also checking relative to the iSellR controls, you can maybe clean it up a little bit better as well.
Yeah.
But I think also a problem here is just visualization. With the red it's hard to discriminate that these are not as red as these.
You could set the scale for that too.
Yeah, you can adjust that.
Because I think it's
And this is all stuff we already talked about. Just to reiterate, these are some of the things that have been really useful for me in iCellR, and I've done all my CITE-Seq analysis in this package. So.
And I think iSellR is just, I think the real power of it, maybe it holds here, is for VDJ. Does a through analysis for single cell VDJ analysis.
Mm-hmm.
Probably.
I don't have anything about VDJ analysis yet. Yeah, that's really it. You want to go back to where?
Can we combine this with on frozen samples if we have to do like single nuclear sequencing?
On frozen sample, I'm not sure if it's been. I don't know. I don't think so.
You saw them and then you add the antibodies?
Well,
Let them dissociate the cells and then
Well, first of all, I would dissociate before you freeze though.
Oh, okay, wait, so yeah, we're talking about frozen.
Age-old frozen samples which people used to do
Doesn't- yeah.
nuclear sequencing.
It doesn't work very well. If you first associate, then you freeze it slowly in DMSO, you can do it. But then it's an interesting question of
Not
it's still
tissue
the site-
sections though, right? We're talking about cells in suspension.
So in clinics, usually there are fresh frozen tissues from like 1960s,
Yeah.
which we can thaw, and then people do the nuclear sequencing, the single nuclear sequencing,
I don't know if it works.
because that works.
I don't really have an.
So
Yeah.
we've done it in our lab on frozen bone marrow samples and it works.
Single cell?
Yeah.
And it works?
Yeah.
Oh, so say more.
Um,
How'd you do it?
so
Single,
yeah, we just did like frozen bone marrow. We had Alireza analyze them. We had normal bone marrow.
So you thawed them and
Yeah, we thawed them.
you still had single cell?
Yeah, same process like we would stain for cats.
so it's nuclear sequencing or
No,
single
no,
cell?
Were they alive?
Yeah, they were like, as long as you have
But I
viable
think his
cells.
samples from
Oh, the
1966 are
frozen,
not,
the frozen tissue,
chances
for example,
are.
kidneys or muscles.
So I think as long as they're dissociated and viable, it should work.
Yeah,
How do
they
you
have to be
freeze
They,
viable.
how would they be
That's the
viable?
bottom
So they were
line.
dissociated before
Yeah,
you
they were
froze them.
dissociated.
So it wasn't like just a complete bone marrow.
No.
Oh, you
No,
dissociated.
so I think-
Oh,
Yeah.
dissociated.
Oh, yeah, that one. I thought you said you
Right.
basically thawed the tissue. That, yeah.
I don't have any
So no one, does
experience
anybody
with
have
this.
experience here with thawing tissue, not dissociated? Yeah, because that, I would think that's much harder.
Yeah, I don't know. I mean, all I can say really is that if it works for single cell RNA-Seq, I don't see any reason why it shouldn't work for CITE-Seq. But whether it works for single cell RNA-Seq, that I don't know.
So, yeah, frozen people don't do single cell, they do nuclear sequencing, single nuclear sequencing.
Well, there is also
Cells are not possible.
this, the single cell ATAC-Seq technique is starting to be used a little bit more, which is only on nuclei. I'm actually gearing up to try it soon, but it seems to be working. Not too many people have done it yet, but 10x has a kit for it. So there may be some way to kind of think about how that works and use it for what you're talking about.
No, I just theoretical question.
Yeah.
All right, thanks, guys. So I think at this point, we should, I should just say that next time we thought we would talk about how to smooth your data, impute data. There's methods that have been developed here. And now we thought we'd give you guys some treat, and you're all welcome to stick around if you want. And talk to Reza, talk to Joe, talk to whoever's here, and just talk.